In this video, we'll be looking at plant stress responses towards abiotic stresses. So, namely, there are two specific ones that you need to know, which are their response towards a change in the daylight. So usually that's when there's a change in seasons, and the other one is usually when there's a lack of water. So the first one we'll look at here is a change in daylight. So we're not talking about the change in daylight throughout 24 hours. We're talking about seasonal changes. So when the plant is actually detecting that over several days or months that actually the day is actually becoming shorter. So this is when we're thinking about transitioning in autumn time or in winter. So here we can look at this process, which is called abscission. Now, abscission is a particular process where the leaf gets cut off, right, from the leaf stalk. So it goes through a particular process. So here, let's say, for example, you can see there's the main stem, and then we've got the branch there that is connected to the leaf petiole, which is basically saying that there's a leaf there. So we're going to focus actually on seeing what is actually happening in this particular part there at the base of the leaf stalk. So the first thing is there must be something that is detecting the change in the daylight. So the first thing that happens is that to be aware there's a particular light receptor called phytochromes. So phytochromes are basically light receptors that you would expect to see, for example, like the ones that you see in your eyes. Basically, they are actually these particular receptors that control seed germination, flowering, and obviously, as we can see here, in abscission. So there are actually certain research that shows that an active phytochrome can actually act as kinases and interact with transcription factors. And from there, they can actually affect the genes that are related to the production of auxins. And that becomes important because we can see how the auxin production is then altered and how it affects other hormones there. So this is potentially a link to the other chapter about transcription factors and the control of gene expression. So just keep in mind that this is a particular example of a kinase that could interact with transcription factors. So basically, once they have detected a fall in the light levels, what they would do is, as I mentioned, is that they will then actually affect the level of auxin production. So in this case, it actually decreases the auxin production. So it stops further auxins to be produced, usually from the stem cells. And from that, in that sense, once they have a less auxins, the leaves would respond by actually producing more ethene. So they would start increasing the ethene levels in the actual leaf. So once the ethene is made, it will actually kind of diffuse and spread down towards the base of the leaf stalk, which you can see here that is in pink and yellow. So this whole area is called the abscission zone. So the point is that the cells that are actually found in the abscission zone are quite sensitive towards ethene, so they will respond to ethene. So what actually happens here is that the ethene will then actually go into the cells and switch on the genes to produce enzymes. So the ethene activates the genes in the cells that is within the abscission zone to produce digestive enzymes, and as the name implies, the digestive enzymes will then go on to digest the actual cell wall. So the enzymes will digest the cell wall in the yellow area, which is called the separation zone, or the separation layer, because that's where it actually falls off. So in that particular part, and also just kind of to expand our understanding, remember that the plant cell wall is made up of cellulose, which is a type of carbohydrate made up of beta-glucose. So keep in mind that the enzymes that could digest the cell wall is actually about how the enzymes or enzyme cellulase could actually digest perhaps the cellulose in the cell wall. So just to stretch a little bit more towards the other bits that you would learn in biochemistry. Then basically, finally, the last bit is you can see there's a pink area there. So the pink area is what they call as the protective layer. So what happens here, keeping in mind that in the stem there would be the vascular bundles, the xylem and the phloem, etc. And what happens here is that the vascular bundles in the pink layer here would be sealed off, right? So it's kind of like clotting or forming a scab when you get hurt in animals. So this bit, the vascular bundles get sealed off, and also there might be fatty deposits as well, so kind of like scarring. And the idea is that to make sure that any of the chemicals that are transported in the xylem and phloem won't be allowed to kind of escape or leave via that particular wound, so to speak. So this is the final bit in the pink area. Right, so the vascular bundles are sealed finally, and once they've done that, with the help of other factors like the wind, as the wind blows, the leaf will then fall, because at that point actually there is already a bit of a separation between the two areas here and the abscission zone. So that is how abscission occurs, especially when there is a change in the light levels. And why might this be important, you might think? Then actually, if you think about it this way, in the winter time there is a shorter daylight, meaning there's less light available for photosynthesis. So the plant would actually go ahead and think, okay, if there's less light there, that means there's not that much point in actually making sure they can produce the chemicals for photosynthesis, right? Because it does actually require a little bit of energy to make sure everything works. So, and not to mention it's going to be quite cold, and they don't want to freeze off the roots on the ground. So the phytochromes detecting the falling in light levels, or in some sense detects the change in seasons causing abscission to occur. The leaves fall off, meaning they don't do as much photosynthesis, because there isn't much light anyways, so you save a bit of energy to maintain the leaves. And also the fallen leaves on the base of the tree trunk can actually form an insulating layer, and it kind of keeps and prevents the roots from freezing over during the winter time. Not to mention that eventually they would rot, and that provides nutrients back to the soil and provides the nutrients for the plants to reabsorb. So that is abscission, which is a stress response towards falling in light levels. Another thing that stresses plants out is a lack of water. So when the plant isn't really being watered, or there's little rainfall and there's not enough water in the soil, underneath the soil there, then one thing that the plant will try to do to retain the water or to reduce the water loss is by closing the stomata, right? Because that's kind of the main way where water can be lost via transpiration out of the stomata. So by closing it, they're reducing the chances of water loss. So if there's very little water in the soil, the root hair cells will detect that. And once they have detected that, they will start producing these chemicals, this hormone called abscisic acid, ABA. And ABA, once it's been made, will then be transported up the roots, up the stem actually, and to go towards the rest of the plants, especially the leaves, because that's where the guard cells are. So as the abscisic acid travels up the stem and it goes into the leaf, it will then bind to these complementary receptors on the surface of the guard cells. And the guard cells are actually coupled with particular ion channels, as you can see here. And once they bind to it, it will cause a conformational change, and the ion channels will actually open, and it causes the ions to diffuse out. You can think about it in terms of how the guard cells are now losing the ions, meaning their water potential goes up. Or you can also think about how the ions are now out of the guard cells, meaning that the water potential outside the environment is actually lower. So outside, there's a lower water potential compared to inside the cell. And hence, the water, and you can see in the blue dots, the water inside the guard cells, which is what makes it turgid in the first place, would then leave. Right? The guard cells will then allow the water to actually escape from the guard cells themselves, and hence, if the plant cells are losing the water, they lose that turgor effect. Right, and therefore they would go from being turgid to flaccid or even plasmolized, and then hence they would kind of go inwards and shrink in size, hence closing the stomata. So to summarize the response of plants when there is little water, and you can see in the first part here, the roots would detect a lack of water in the soil, and they would then produce abscisic acid, ABA. The abscisic acid then travels up the stem into the leaves, and it binds the receptor. You can see on the top here, it binds the receptor there on the guard cells, and that would cause a change in the ion channels, which is as represented by the box here. And once they've done that, the ions will then move out of the guard cells, as represented in purple here. And because of that happening, it will then affect the water potential gradient in and out of the guard cells, and therefore the water would also leave the guard cells by osmosis down the water potential gradient, and because of that the guard cell loses its shape and become flaccid or even plasmolized, and hence kind of closes in and causes stomata to be closed. And remember, the reason why closing the stomata will help is because it would reduce the transpiration rate, meaning that you are potentially stopping water from being lost from the leaves themselves. So you're therefore retaining more water. Notice that this is not exactly helping the plant to absorb more water, but it's simply about trying to help the plant retain the water that they've got so that they don't become further dehydrated. And these are the two plant responses towards abiotic stress.